The decoding of the human genome in 2000 ushered in a new era in medicine. A pioneer in genomic medicine, Phalanx Biotech Group is one of the 4 prominent global DNA microarray developers and manufacturers, and the only OEM high-density microarray biochip firm in Asia. Hailed as Asian biotech's health hero by the Discovery Channel, the company was featured in a special interview in America's Fortune magazine, speaking on behalf of Taiwan's biotech industry to an international audience. Located in Hsinchu Biomedical Science Park, Phalanx Biotech is Taiwan's only microarray firm with independent gene microarray development technology. Phalanx Biotech's formidable R&D have inspired the publications of several hundred essays in international journals and the development of customized scientific genetic testing methods. It also offers various advanced genetic testing technologies, such as gene microarray biochips, next-generation sequencing methods, and big data analysis. Phalanx Biotech has earned major biotech R&D awards and received over 50 patents in numerous countries. In the future, its R&D center will continue to develop cutting-edge microarray technologies and broaden their applications. As it pursues independent R&D capabilities and provides stable, reliable products, Phalanx Biotech received the Taiwan Accreditation Foundation's ISO/IEC 17025 laboratory certification and is applying for LDT laboratory certification. Phalanx has received GMP and EN ISO 13485 certification for its microarray and reagent plant and is applying for product IVD certification for overseas expansion. Cherishing life is Phalanx Biotech's core principle, and every one of its gene microarray testing methods puts people's needs first. From newborn health to preventive medicine for adults and precision medicine to treat cancer, Phalanx offers a wide range of testing applications. Phalanx Biotech continued to make improvements after launching its ACGH chromosome microarrays in 2015. The Phalanx amniotic fluid microarrays can screen fetuses for over 500 types of hereditary and congenital disorders caused by copy number variation, including growth retardation, mental retardation, and organ dysfunction. The microarrays can achieve a detection rate 4 to 5 times that of conventional karyotype testing. Phalanx's amniotic fluid microarrays are the sole chromosome microarrays in Asia able to detect whether a fetus carries the gene for alpha thalassemia. Phalanx's microarrays can detect the problem even when a fetus has the slight CNV abnormality causing Duchenne muscular dystrophy, DMD. Phalanx Biotech is also developing many next-generation products, including unique patented electrically controlled microfluidic technology to acquire intact cells from a fetus, expected to be used in non-invasive whole genome prenatal testing in the future. With funding from the Ministry of Economic Affairs in 2016, Phalanx Biotech developed a next-generation sequencing method that can screen for growth and mental retardation in children ages 2 to 6. Phalanx is also a leader in developing dual-platform testing, employing integrated microarrays and WES, used to detect CNV abnormalities and single nucleotide variant SNV's mutations. This solution helps doctors identify whether the cause of illness is hereditary or congenital and provide proper treatment. Genetic research conducted over the past 20 years has found that everyone's state of health depends on their genetic endowment. Genetic information can thus be used to draft individualized health management strategies. Phalanx's SNP Health Risk product has unique modes of operation for both congenital and postnatal risks. The high-resolution dual allele probe design has also used its qPCR platform to develop genetic tests for deep vein thrombosis, osteoporosis, skin health, etc. These systems give people a better understanding of their state of health, allowing preventive measures. The patented ctDNA methylation technology we acquired in 2019 can be used to identify the liver cancer methylation index from blood samples and can detect cases of BCLC, 0, liver cancer. With sensitivity as high as 83%, the method is more accurate than the conventional alpha-fetoprotein indicator. Our early cancer screening allows patients to receive early treatment, which greatly boosts survival while reducing medical costs.
Hello everyone, thank you for joining us. I'm Dr. Liu, the principal scientist of Phalanx Biotech company in Taiwan. It's my pleasure to talk to you about the clinical application of DNA methylation in cancer management. DNA methylation is a kind of epigenetic modification in gene regulation, which also includes histone modification and microarray. It occurs on the cytosine of CpG sites. One enzyme family, DNA methyltransferase, catalyzes this process by adding methyl group on the fifth carbon of cytosine bases, leading to the gene silencing. In the progress of carcinogenesis, some of genes will be silenced by DNA hypermethylation, such as tumor suppressor genes and DNA repair genes. On the other hand, some of genes will be overexpressed by demethylation, also called hypomethylation, such as oncogenes. Especially, aberrant DNA methylation will be occurred in the very early stage of cancer formation, even in the asymptomatic stage. That is why DNA methylation is a powerful biomarker for early diagnosis of cancer. Recently, liquid biopsy is a very hot topic in cancer research. We can find many valuable biomolecules in blood, such as ctDNA, ctRNA, microarray, and CTC. Taking the benefit of liquid biopsy in clinical usage, aberrant DNA methylation also can be detected in liquid biopsy and applied it to the real world. There are several advantages of DNA methylation acting as methylation markers in cancer management. For example, aberrant DNA methylation occurs in the very beginning of cancer progression, so it makes us achieve a long-time goal, early diagnosis of cancer. Furthermore, methylation profiles show very specific patterns in different cancer types, implying that methylation markers have a property to diagnose cancer with high specificity. As previously mentioned, DNA methylation can be detected in any kind of liquid biopsy samples, including plasma, sputum, stool, and urine. We use a qPCR-based method to detect methylation markers in clinical samples, which is called QMSP, quantitative methylation specific PCR. It is very sensitive, allowing us to detect very rare methylation markers in plasma samples. Over the past few years, we have established the DNA methylation analysis platform, including whole genome methylation analysis, big data analysis, and various methylation experiment skills. This platform technology is not only suitable for cancer research, but also proper to study in other interesting fields. In the cancer management, from the beginning of screening, diagnosis, prognosis, therapy selection to the monitoring, every stage needs precise biomarkers for the purpose of precision medicine. DNA methylation is very well suitable for this application. The first project is the early screening of liver cancer. We use the methylation platform technology, as I mentioned before, to identify novel methylation markers in the discovery phase. In the preclinical study, we use both a qualitative and quantitative method called COBRA and QMSP, respectively, to validate these markers in the clinical samples, including tissue and plasma. In the next step, we have finished 2 clinical trials. One was cooperated with National Cheng Kung University Hospital. The other was cooperate with 4 medical centers, including Taipei Veteran General Hospital, Kaohsiung Medical University Hospital, Qimei Medical Center, and the National Cheng Kung University Hospital. More than 1,000 cases were validated. Now we can provide testing kits for clinical laboratories or receive clinical samples for the central lab service. According to our studies, the sensitivity and the specificity of methylation markers for liver cancer diagnosis reached to 84.2% and 83% respectively. In the same population, alpha-fetoprotein, the gold standard for the liver cancer screening, shows the 51% sensitivity and 62% specificity. The performance of methylation markers is much better than alpha-fetoprotein. 75.6% alpha-fetoprotein negative patients with liver cancer can be detected by methylation markers. Furthermore, 83% 0.3% liver cancer patient can be identified by methylation markers at a very early stage, meaning BCLC 0 stage. It makes us to achieve the goal of early detection in liver cancer. If you feel DNA methylation is fascinating, please feel free to contact me. Thank you.
Hello, I'm Min, the next generation cell-based pregnancy test project leader in Phalanx Biotech. We are excited to announce our tremendous progress in the development of this product. In Phalanx Biotech, we would like to provide a prenatal test total solution based on the different requirements to offer the best prenatal test package. We provide ACGH, cyto-OA array, and the NIPT service now. Moreover, we are developing a new technique to provide an alternative option for the customer, the non-invasive cell-based prenatal test. We know the major commercial product prenatal test contains invasive method like detecting the gene disease from infant cell through array-based DNA microchip, and get the mother's blood to analyze the possible chromosome issue from cell-free DNA of the infant. In Phalanx Biotech, we want to provide an alternative prenatal test option for the pregnant woman can select with a more precise testing result than cell-free DNA-based NIPT and a more safety sampling method than the amniocentesis. Therefore, we are excited to announce that our tremendous progress in developing these products by a bioelectronic chip to get the third generation prenatal test result. Currently, NIPT can detect 10 to 100 gene disease, and ACG's DNA array can provide the diagnostic result on all the major gene diseases. Our preliminary data shows that we are possible to get the whole gene from our enriched cell sample. In here, I'm going to summarize our next generation prenatal test. Our bioelectronic microfluidic chip can enrich the infant blood cell and provide enough cell from whole gene screening on the gene disease detection. Our test will first offer the same performance as the current method to detect the trisomy 13, 18, 21. Second, we want to provide more than 300 CMV disease and the single mutant detection result. Most important, we want to offer the test in the first trimester as early as around 7 weeks to 12 weeks of pregnancy. In addition, we can directly assemble the polymer inside of a chip to achieve a high yield. Based on our chip design, we can accept the wide variety of the different clinical sample conditions. We also provide solution for the cell isolation technique on eliminate non-specific trap of the simple sample by high flow rate, reduce the channel clock rate by the design of chips. Third, isolate enough amount of cell by our bioelectronic system. Shorten the microfluidic operation time to speed up the progress of the experiment. The most important function of our chip is we can batch capture and release the sample to save the working force. Our bioelectronic isolation chip can provide high efficiency target cell capture and release capability to get tropoblast from the specimen. With this fantastic device, we can finish the cell capture and the isolation within few hours of operating. NGS or ACGH can apply to the isolation cell sample to detect the gene defect by a higher resolution non-invasive prenatal test compared to the current cell-free DNA NIPT method. On the other hand, we offer a reduced cost and time-saving strategy on the stable chip fabrication method and more accessible operating interface for the future service required. Thank you for listening. Here is my contact information. Feel free to ask me any question or send me email. Thank you.